Welcome to this 10th HBP Tea Slides. I am Carmen Lupascu. I'm a postdoc at the National Research Council in Palermo, Italy, and I will be your host today for this event. Exactly 1 year ago, we started this series of events where young researchers from the HBP are presenting their research. There are two 10-minute talks followed by a 10-minute Q&A session. And the two speakers from today are Roberta Maria Lorenzi from University of Pavia and Simone Russo from University of Milan. Before we start, if someone experiences technical issues, I hope that you would not experience technical issues. But if you do, and the issues cannot be solved during the session, please contact the education team at the email education@humanbrainproject.eu so they can try to fix the problems for the next session. Also, you should post your questions in the Q&A chat. There's an icon on the bottom, and questions will be answered after both talks. It is also possible to use the raise hand function, and I encourage you to do so. There's an icon on the bottom. And ask your questions verbally when unmuted and called by me. The recordings and the presentation slides can be found in the education program's e-library a few days after the event. After this session, we can continue the discussion in the HBP student Slack channel. An invitation link will be posted in the chat. So I think we can start. Our first speaker is Roberta Maria Lorenzi. She holds an MSc in bioengineering at the University of Pavia, and now she's a PhD student at the Neuroimaging and Neuroengineering Lab at the University of Pavia. She's working with the Computational Neuroscience Research Group of Professor D'Angelo, and she's focused on the development of a computational model of the cerebellum. The main purpose of her work is to develop a model able to reproduce the cerebellar activity, which could be integrated in the available simulators such as the virtual brain. So Roberta, if you are ready, you can share your screen.
Thank you. So, hi all, and thank you, Carmen, for the introduction. Today, I'll introduce to all of you our mean field model of the cerebellum. So, the background of our model is the brain, which is made of interconnected networks, and these interconnected networks generate the global activity of the brain. Cerebellum is a brain region, and it contributes to generate the brain global dynamics. There are a lot of approaches to investigate the role of local network in brain dynamics, but there is no specific model for the cerebellum. If you are wondering why we need a specific model for the cerebellum, the answer is that the cerebellar circuit organization is very, very peculiar with specific cells populations, and it physiologically differs a lot from the cerebral cortex circuits. So in this context, we aim to develop an advanced mean field model of the cerebellum with a double purpose. First of all, we want to have a tool that provides us an insight on the cerebellar network functioning, and a model as flexible as possible in order to be integrated in available tool like the virtual brain, that is a whole brain dynamics simulator. Okay, so the steps that bring us to develop our advanced mean field model are, first of all, network design. So we thought about which populations include in our model, and our starting point is a single neuron scaffold model in order to set biological parameters in order to be very coherent with the physiology and biology of the cerebellum, cell populations and its connections. Then we have to translate this network design into a mathematical formalism that are equations, like the one you can see here, where the core is this part, pointed by this purple arrow, that is the transfer function, and that is the core of the mean field formalism. And then we have to make a computational form of this mathematical formalism. So we implement these equations in Python 3.8, and in particular regarding the transfer function, we perform a fitting of the semi-analytical expression of this function that I'm going to explain you better what it is and its role. Okay, so let's have a focus on the network design. The population that we included in our model are the granular cell, the Golgi cells, the molecular layer interneuron, and the Purkinje cells. We considered the connections, excitation, and inhibition, and the functional reference of our model is the spiking activity of neurons that are modeled as edge-leaf single point neurons. So we start from an edge-leaf equation for each of these cells that you can see in the network. Okay, then starting from scaffold model, we set connection probability, the K, you can see here near the arrows, and also the quantal synaptic conductance in order to generate physiological population firing rates. All these parameters are used to define the equation of population-specific conductance, that is the starting point for our mean field model. Here I write an example for the MLI, but the rationale is the same for all the population, and so we have an equation like this one for each of the population of our network. Okay, we used this population, this conductance equation for each population in order to compute the membrane fluctuation properties. In a mean field formalism, the membrane fluctuations properties are a sort of summary of statistical summary of the behavior of the populations that we included. So the population-specific conductance take as input the presynaptic frequency input and also population-specific property as the adaptation current. With this equation, we compute the membrane fluctuation properties that are the average membrane potential, the standard deviation of the membrane potential, and the autocorrelation time. These are the main features to define our transfer function expression through a semi-analytical approach. And this semi-analytical approach is implemented as a fitting in two steps. First of all, we have to fit a so-called phenomenological threshold, and then this phenomenological threshold is used to optimize the expression of the transfer function. In terms of computation, this formalism is translated in a code written in Python 3.8, and we start from the computation of a transfer function numerical template. Then this transfer function numerical template is the input for a function that defines the population conductance, that is this equation here, and then we compute the fluctuation properties. You can see here an example for the Purkinje cells, but the rationale is the same for all the populations. And once we have computed the fluctuation properties, we are able to fit our semi-analytical transfer function expression with the package of Python provided by Python 3.8 that you can see there, the HBP optimized package. Okay, once we have obtained the transfer function, we have completed our mean field formalism, and we are able to write an equation like this for each population. Here the P is refer to all the populations, so we have 1, 2, 3, 4 equations of this kind, and for two populations, for the Golgi cells and for the Purkinje, we have 2 more equations in order to model the adaptation current. With this set of equations, we will be able to describe the activity of our cerebellum in terms of mean field model, in a form that can be as flexible as possible to be integrated in already validated simulator like the virtual brain, but also like DCM, and so to provide to the user a tool that can be very useful to study the dynamic of this region of the brain. I would like to thank you all for your attention, and if you want, it's time for questions and then also for another coffee.
Actually, thank you very much, Roberta. We don't have any questions yet, but all the questions will be answered after the second talk.
Okay.
So feel free. I remind the participants to post their questions in the Q&A chat, and we'll answer the questions after the second talk. So now it's time for the second speaker, Simone Russo. He graduated from the University of Milan in medicine and surgery, and he's doing a PhD in mind, brain, and reasoning with Professor Massimini. He mainly works with non-invasive and invasive stimulation and recording electrophysiological techniques in humans to study the physiology of consciousness, wakefulness, sleep, and the pathophysiology of disorders of consciousness, focal brain lesions, and epileptic disorders. So, Simone, you can share your screen.
Okay, well, whenever we think about focal brain lesions, we imagine just to remove a bunch of neurons. Yet, in network theory, it is well known that removing one node can have larger detrimental effect at the whole network level. Similarly, in clinical practice, we frequently observe that focal lesions may have distant consequences, such as symptoms are related to the lesion site. Our work demonstrates that focal brain lesions do not just knock down a bunch of neurons. They interfere with the functional integrity of the entire brain. To demonstrate this, we take advantage of a unique clinical setup based on patients diagnosed with drug-resistant focal epilepsy that undergo a minimally invasive neurosurgical procedure, that is the implantation of depth electrodes or stereotactic electrodes. Whether the diagnostic investigation is successful, those patients undergo a minimally invasive neurosurgical treatment that is called radiofrequency thermocoagulation and perform a small lesion in the target site. We will shorten radiofrequency thermocoagulation in RFTC, otherwise we will need a longer time slot. In panel A here, you can see the MRI of one patient together with the reconstruction of his brain surface. The 5 white dots illustrate 5 intracerebral contacts employed to record the activity from deep and cortical structures. Besides the white dots, you can see the black ovoid that depict the radiofrequency thermocoagulation site, that is the lesion. In panel B, you can see the traces recorded before the lesion in each of these contacts in black, and after the lesion in gray. As you can see, the rapid, the synchronized rhythms typical of wakefulness are replaced by slow rhythms that are typical of sleeps. This effect is maximally prominent in T4-5, that is the contact nearest to the lesion. Thus, for each of these contacts and conditions, we computed the power spectrum in panel C, and then we computed the variation of delta power that you can see in panel D. Traces, spectra, and delta quantification suggest a trend showing that the increase in delta power is maximal near the lesion and then decays with distance. To confirm this empirical observation in a larger population, we explored 21 patients with different lesions and explorations that you can see in the flat map on the left. In so doing, we found that the delta variation depicted on the y-axis on the right, we depicted the delta variation as a function of distance that is on the x-axis on the right panel. And we found a steeply decaying trend that that confirm that this effect is maximally prominent near the lesion and decays with distance. Well, we performed a cluster analysis, finding that there are two groups of contacts. One is near the lesion, and we called it hyperlesion because it suffers from the main electrophysiological impairment. And then, going far from the lesion, we have most of contacts that show almost no difference in their delta power. Yet, there are distant contacts that show somehow an high increase in delta power. And thus, we asked whether these distant contacts showing high increase in delta power reflected a preferential connectivity with the lesion site. Thus, we computed the connectivity with the lesion as the power of the response to the stimulation of the RFTC site itself, that you can see in red in panel B. Together with the connectivity, we evaluated the distance from the lesion itself in green and the variation of delta power in gray. And we found that in the hyperlesion, the delta variation is maximally correlated with the distance from the lesion itself, while out of the hyperlesion, the delta activity is correlated with the connectivity with the lesion site. In physiological conditions, the only case in which we observe such an increase in delta power is during sleep. Thus, we asked whether this post-lesional dynamic may be somehow related to sleep. Thus, we took the distribution of delta activity during sleep in panel A, and we looked for contacts that showed a similar delta increase after the lesion in panel B. We defined those contacts sleep-like in terms of power of delta activity. But the increase of power that in the delta range that we observe during sleep is not a standalone event, but is associated with the emergence of typical graph elements that are the sleep slow waves. Thus, we asked whether we could find similar graph elements in the post-lesional activity. And we found that the number of sleep slow waves increase in the post-lesional activity with respect to healthy wakefulness. Finally, sleep slow waves are associated with a peculiar neuronal pattern, that is the interruption of the neuronal firing. And we asked whether we could find similar patterns in the post-lesional slow waves. Thus, we performed a wave trigger average time frequency analysis, finding that both in the no-REM and after the lesion, we could find a suppression of the high frequencies that is a proxy that is usually employed to identify off periods or periods of neuronal silence in LFP. And thus we confirmed that both no-REM slow waves and post-RFTC slow waves are underpinned by the same neuronal pattern. There is an increasing number of evidences that sleep can intrude in wakefulness in physiological and pathological conditions. In brain-injured patients, slow waves can explain why perilesional and distant areas become dysfunctional. Crucially, the pivotal role of sleep slow waves in plasticity can also account for the recovery of lost function after the lesion. Understanding the role of slow waves in the post-lesional activity will provide a unique insight on the pathophysiology of focal brain injuries. But furthermore, it will lay the foundations to new therapeutic approaches to treat and rehabilitate brain-injured patients. Thank you for your attention.
Thank you very much, Simone. Very nice presentation. While we are waiting for many more questions in the Q&A chat, may I ask you, Simone, if you had the chance to analyze also the improvement of the epileptic seizures after the thermocoagulation, or?
Yeah, we did also that analysis, and there is an improvement in the seizure, and there is an improvement in the symptoms of the patients, but it is strictly related to the type of disorder of the patient. So it depends, let's say.
But your patients had disorders in this study? How many patients do you analyzed in this study?
21, and all of them had an epileptic disorders, and the implantation is due to the epileptic disorder. And in most of them, there was an improvement in the seizures.
Do you have a percentage of improvement?
It is graded, but we have almost 15% of improvement of total improvement, and then we had different patients that show different kinds of improvement.
I see. And may I ask you too, how many thermocoagulation sites you had per patient? Because when you speak in your talk, it seems that you had only one, but
no, it's not one.
More? Okay,
it's more than one, because the main target is the treatment of the patient. So
yeah, I imagine. So do you have a mean of the
10 sites?
Because I'm a mathematician and I'm interested in
it's something like 10 sites per patient, but we have patients with just one site and other with higher numbers.
Mm-hmm. I see. Very nice. So some questions are coming in the Q&A session. So Harry is asking, do you know how long the sleep-like activity lasts? days, weeks?
Well, we had the chance to record just some minutes of the activity because of the risks of infection, but we are performing further studies with scalp recordings that will answer to these questions on the long term.
I see. One other question for Roberta. Can you clarify the difference of the used advanced mean field model to the standard mean field model?
Yes, of course. In a standard mean field model, we have a Hodge statistical summaries that results into two populations, one excitatory and one inhibitory with the respective connections. In our model, we separate the different cellular populations. I mean, for example, if you look at the granular cell, the excitatory comes from MOSI fibers and has parameters specific from MOSI fibers, while inhibition from Golgi cells with specific parameters of Golgi cells that are not the same, for example, in the case of Purkinje that are inhibited by molecular layer interneuron. So the term advanced and biologically grounded is in terms of parameters and connections considered in our model. This is the main idea.
I hope we clarified, we answered the question. And may I ask you, you said that your model was integrated in the simulator TBP. Can you give us some more details?
Yes, of course. Can I share my screen just a moment to?
Yes, of course. I think.
Perfect. Okay. So, okay, this is the TBP framework, which starts from input coming from structural and functional MRI, and then the virtual brain try to simulate the signal coming from a different brain area. In this setup, there are a lot of models, a lot of different models, but there are not still region-specific model included. So in this framework, we are focused on cerebellum, and we aim to be able to integrate our mean field model as specific for the cerebellum. In the meantime, other research groups are working on the same rationale, but for other regions, for example, hippocampus, basal ganglia, and so on. So the main idea is to have a simulator with a model specific for each brain region, and at the moment we are focused on the cerebellum. So I hope.
Of course, at the end you will connect all these models, yes, even if they are for different brain regions. Okay.
Yeah. The goal. The final goal.
Good luck. Okay.
Thank you.
One other question by Arpit. How the mean field model of cerebellum is compared with real physiological cerebellum?
Okay, we start from a GLIF equation that are a model for a single point neuron, so where the different neuron, granular, Golgi, Purkinje, and molecular layer are considered as a single point with a firing rate. This model, that is the base of our mean field, is validated from a scaffold model implemented in neuron, which considered all the characteristics, the anatomical characteristic of the cells that we considered for the cerebellum. So in this is the pipeline that where the scale grew up from micro scale to a sort of mesoscale, and so we, as a final goal, we will compare our result of mean field with the result coming from microscale in order to validate our biological realism.
So may I ask you if maybe in the future the goal is to use also detailed models instead of single point?
yes, at the moment there are ongoing work for a co-simulation in the virtual brain that is macro scale with single point neurons that is a sort of micro scale, so nest with the virtual brain, and maybe we can do the same once our mean field model is validated and integrated in the virtual brain, just for the part of cerebellum or cerebrocerebellar loop. It depends on the, yeah.
Can you give us also more details on the optimization of the conductances? I'm interested in this aspect too. So maybe you have some more details.
Yeah, about the optimization of you mean the parameters,
yes.
the connectivity, and okay. This optimization coming from already validated and published work. So if you are interested, I can give you the references, and we take this value and try to tune for our model. I mean, for example, for MLI, molecular layer interneuron is composed by basket and stellate, and for a scaffold model, we have basket and stellate not considered together, but divided. And now we are trying to tune these parameters in order to consider one populations, not to replicate something just validated and that works. So it is a step for a bigger scale, we can say. And yes, for the moment, it's a sort of a trial and error procedure, but in the future I hope to find a pipeline to do that in a more engineering way.
I see. Maybe we can continue our discussion on the Slack channel and keep in touch with all these details. Before we close, Simone, question by Alper. Can you summarize why you looked at the delta power? Would you assume similar observations with other spectra?
Okay, the short answer is no. The long answer is that after lesions, the emergence of delta activity is well known from many years, and also before neuroimaging, it was employed to localize brain lesions. So the emergence of delta activity is strictly tied to focal brain lesions from since many years ago. But we have modifications in different bands that are not stereotypical. So there are changes in the oscillatory peaks of several bands and everything else, but the delta band is the only one that behaves always in the same way across all patients, conditions, areas, and everything else.
Okay, thank you very much for your answer, Simone. And as I don't see any other questions in the Q&A section and no hands raised up, I would like to remind all the young researchers from the HBP that would also want to present their research during these HBP Tea Slides sessions to please get in touch with the HBP Education Program at the email education@humanbrainproject.eu. I would like to remind you the next Tea Slide session will be on Thursday, 6th of May, at same time, 2 p.m. And we'll have a talk on how to write proposals for HPC cloud and storage resources. And in the same time, I would like to remind you that registration is still open for another interesting HBP education event, Young Researchers event. It will be on 26th, 27th of May. It will be a 2-day virtual conference where participants can explore how eBrain supports the next generation of brain medicine. So please register. And after the webinar, a browser window will pop up with the survey. Please answer these questions so we get some feedback on the event format and how we can still improve it. It takes only a few minutes. I would like to thank the speakers and see you at the next TN Slides in May. Thank you.
Thank you.
Thank you.
Bye-bye.
Bye.